This video is going to show a full MRM experiment in 5 minutes. This includes picking transitions and comparing results with collected data off an LC-MRM setup. One way to load data into MrM is to load it directly from existing MSMS search results. Here we're loading 3 mascot.dat files all at the same time. I'm clicking on the first, holding down Shift and clicking the third, and all 3 appear. There are options for prioritizing how transitions are picked and for prioritizing which peptides are loaded. All 3 files are then loaded, and you may review what was loaded, including proteins and peptides. The data is interactive, letting you inspect it, filter results, and decide what you want to have passed on to MrM for its initial transition list. You can see the set of transitions loaded into MrM by going into the Current Transition List tab. So here's the list, and you'll see that both B and Y ions are selected. If you want to manipulate how this list was chosen, you can go into the Options tab and change all sorts of things. Here I'm going to turn off B ions because I only want to see the Y ion series for my Q3 masses. Now you'll see only the Ys are shown, and if I control click I can filter to show just that peptide. You'll note that the transitions, the B series, are not gone now, they're just hidden under candidates and filtered transitions. I can toggle these back on, and here I'm showing in the table, under the comment section, reasons why the transitions weren't used. So the B series was filtered by the options I just changed. Showing specific peptides like this and filtering the table with control-click options all use the table value filter down here. You can manually change this, manipulating the table to find exactly what you want to see. When ready to export the results, click on the Export Transition button and type in the name of the file you want to save the results to. This will give you a comma-separated value file, which can then be loaded directly into most instrument vendor software packages. For this experiment, we ran the transition list 4 times each on 2 different samples, a 3-day-old human lens and a 93-year-old human lens. Those results are then loaded directly back into MrM under the Import-Export menu option under MRM data. Again, note I'm going to load all these files at the same time by clicking the first, holding down Shift, and clicking on the last one. The files are already named with the sample types in them, 3-day-old and 93-year-old, so MrM is going to load them automatically as replicates. You could go into the Data Management tab, though, if you wanted to manually name samples and replicate information in there. Under the Transition to Review tab, you can see the transitions. The first is not very good. I can look at the individual replicates and look at another one to verify that it is oh so bad. And then there's an interpolated view that will show all replicates at the same time, which is a convenient way to note that the whole thing looks bad. I can then go in and toggle off any of the transitions for this Q1 family of transitions, and there's a Reject All button that would do the same thing. This next transition actually looks good here, so I can go into the table and adjust my signal calculating bounds, baseline noise, and things like the mean retention time, which is helpful if you're doing scheduling later or exporting the data for use outside of MrM. The next button accepts it, and I can continue on reviewing transitions, accepting, rejecting as appropriate. It is important to note that these graphs are fully interactive. You can manipulate the data, zoom in, zoom out, and generally inspect it in any way you would want. If you want to look at a particular transition family, that is all the Q3s for a certain Q1, you can go into the Find field and just type it in. And so here I found one, and I can go in and continue to edit it, just like I was doing with the other transitions. When you're done manually reviewing transitions, you can go to the Visualization panel to see an overview of all the data in your data set. So the scatter plot is showing us coefficient of variation versus log intensity of a transition set. And you can see there seems to be a link between higher intensities and lower coefficient of variations. The graph is interactive, so I can click on a point to go and see the actual signal for it. And then when I'm here, I'm just on the Review tab again, so I can adjust the UC bounds as I like. And back under Visualization to do this for any of the data points for my data set. These figures can be exported for publication and things of that sort. There are many other plots in here. Here's one that's a histogram showing the coefficient of variation across the whole data set. Interactive also means these plots are zoomable, so here I can zoom in and see previews of the signal calculation for each of these data points. And so here I'm looking through them, and I can click on them to go to that Transition to Review tab and further manipulate the transition if desired. Okay. After doing QC for the data set, I can go in and actually look at changes between the 2 samples. So I have the 3-day-old and 93-year-old here. At first I'm looking at all the data, so it's kind of a mess. But I can go in under the filter and type in show me all peptides that are deamidated. In this data set, the hypothesis deamidation causes a hydrophobicity change, which is correlated to cataracts in human lenses. And so here we can see in the 93-year-old sample, there are many cases where deamidation is quite a bit higher than in the 3-day-old sample. So I can click on the chart, say show me just those transitions, and then go down and export that data for use in Excel or further data processing as I desire. That is the whole experiment in 5 minutes. Be sure to use the link and contact email below for more information, and please contact us about licensing Mr. M or asking about help with your MRM data analysis.